doctoral associate in Dr. Rodriguez Martinez lab. And our department lies in the intersection of biomedical information, informatics, and data science. And we are affiliated to Yale School of Medicine. For today's presentation, is basically talk about our recent archive paper focused on benchmark study to review this generative models for the scaffold designs, especially for the complex for the peptide TCR receptor peptide MHC complex. So I would like to thank you to the computational intelligence group for hosting me today. And we are going to dive into this fascinating intersection of generative artificial intelligence and computational immunology, which our group are focusing. So specifically, we will discussing the de novo design of the peptide as the T-cell receptor and peptide and the major histocompatibility complex interface. So we're known as the TCRP MHC complex. As a computational intelligence researchers, you are likely familiar with the explosions of the generative models like diffusion networks. So today we will explore how this state-of-art diffusion models perform a highly constrained, geometrically complex biological environment. So we will walk through a novel multi-step computational pipeline we designed to benchmark and optimize the peptide predictions. So before we discuss the algorithm, let's briefly ground ourselves into the biology. So we know that T-cell receptors recognize the antigenic peptides that are presented on the surface of the cells by the MHC molecules. So MHC is a short notation for the major histocompatibility complex, and you will hear this notation a lot during this presentation. So just take a note over here. And additionally, think about that every cell in our body as having a window to display. So it's constantly take a small piece of the protein it's making inside the cell and into this window for the immune system to inspect. So this window displays actually more like form this kind of status report or cell barcode so that T-cell receptors can scan it. So our immune system actually have this kind of patrol guide, it's T cells, and that can constantly scan this barcode. And they use this special scanner, you know that this T-cell receptors form as Y over here, and you will see this shape in this presentation several times. And use this T-cell receptors, they can check the status report of the cells. So if these cells return different signals, so say that health cell will show the normal proteins, and then this T-cell receptor will give its pass. But for some disease cells, it will display some abnormal proteins. For the cells, cancer mutations or viral proteins, and this will get destroyed by the T cell, and it will initiate the adaptive immune response. So on the right, this is actually the T cell looks like at the molecular level. They are really reading the peptide, and you will see that the peptide is highlighted in this box, and it's grouped by the MHC complex, MHC content, and then it's touched by the T cell receptors. So during this, for additional information, so you might know that during the T cell development, the cell will randomly select this V genes, D genes, and J genes from each of this region, and they combine them together. Because it's the random combination and have this nucleotide mutation, it will form kind of the millions to 100 billions of different T cells. So that means in our human body, we have much more T cell receptors than T cells. So as I provided enough information for this immunology, I would like to walk through of these protein structures. And we know that protein folding is more like the original puzzles. It solves by itself. So if you have a long string of amino acid sequence like this, it will automatically fold into a 3D shape. So for the second level of structures, you will notice this is the alpha helix and the beta sheet. And the alpha helix is more like a spherical staircase made of the amino acids of dozens make of the dozens of amino acid, and it's one of the most common structures in the protein, and that's protein will naturally fold into. And for our TCR receptors, you will observe several alpha helix in the shape. So talking about the molecular checkpoint, this three molecular interactions, the TCRs, TCR receptor on the top, and peptide in the middle, and the MHC located on the bottom. And this is the central checkpoint of our human adaptive immunity. It will decide whether our immune systems ignore the healthy cells, as we described previously, or attack virally infected or cancerous cells. Or for the autoimmune settings, they will misfire against our human body itself because of this dysfunctional the peptide. So from a therapeutic perspective, if we can computational design and discover this specific peptide located in the in the middle, so we can dive into the next generation of the cancer immunotherapy for the customers of vaccines in the future. So since we have seen how T cell naturally scan for cellular status, but here is another problems. So natural T cells receptors have major limitations. They are real and hard to isolate from the patient. So it's often not specific enough for the therapeutic use. So for the decades, it has been a bottleneck in the immunotherapy domain. So in the recent years, the AI revolution has changed, or I'll say transformed the current situation. In just 4 years, we went through the predicting existing protein structures from the AlphaFold 2 to designing the entire new protein structures from scratch. So that's bring the innovations for our today's work. So I actually got inspired by this pipeline. So use this AI tools that's create entirely the new peptide that can be recognized by the TCR and MHC. So I would like to show you an example over here. So this is the animation to show the AI guided protein design. You will see that this blue structure is the alpha helix, as we mentioned previously. And this helix can dramatically change the position during the AI diffusion steps. And this protein will undergo the precise conformation that change its location that can be bind to the protein as your target. But still for current phase, there's still a massive gap in this field. So we actually currently lack of a systematic benchmark to evaluate how these generative models perform in this high restricted constraint into the TCR MHC domain or TCR MHC interface. So let's bring our study to this benchmark. So for the first stage as our study, we need to find how to model the TCR receptor and the peptide MHC complex. So because it's a very highly dynamic process, and also it's highly dynamic complex. So model this three tripods is really incredibly difficult. So we should tear down the whole part into different combinations. So they will start as analyze the peptide and the MHC group. For this part, you will see that there is a geometric constraint. So the peptide is actually not free floating. It can go to different location. But this MHC groove actually constrain the peptide into the B pocket and F pocket. So B pocket is basically a region that's anchor the second amino acid for the peptide, and the F pocket actually constrain the end of the amino acid for the peptide. So if we can define the B pocket and F pocket accurately, then we can constrain this peptide. So say that's it's only generates between this B pocket and F pocket regions. So here, so we know on the left side, if the peptide in the natural space, it's actually floating freely in solutions, and they will existing as the unstructured loops. However, if we provide this binding constraint, this peptide can actually undergo an induced feed, and it will adapt itself into this high specific and constrained conformation. So based on this combination, our ultimate goal is to generate the diverse and physically plausible peptides within this MHC group. So you will see from this whole process for the Rosetta diffusion, which we use for our whole study for the benchmark analysis. This to achieve this complex balancing of the structural freedom and this strict geometric constraints. This alpha Rosetta fold diffusion released by the David Baker team is widely used for the protein binder designs. And for our peptide designs, we actually went through different tools like Bidecraft's Rosetta field diffusion 1, 2, 3, and we end up with only focus on the Rosetta field diffusion 1 and 3 because they can provide a high performance on the peptide designs. And you can see that the first step is that we bring us with the denoting diffusion probability models. And this generative process begins at time step t at t with the pure Gaussian noise. So the whole process will be denoise step by step and finally return a structured shape for the peptides. So you will see this is actually the reverse diffusion process. Once this model has been trained well, we will end up with physical plausible structures for the peptides. So the whole process is illustrated in these 3 steps. So you might wondering how this Rosetta field diffusion works. So basically, it's went through 2 steps. As you can see from this animation on the right corner, so there is the forward path will from the right to the left side, and the Rosetta field diffusion first will learn how to take this real protein into as an input, and then it will systematically add the noise into different steps, and then it will end up with a cloud of the noise at the left. So it's we call this the forward process. And the model have additional reverse process. So the reverse process most important in this structure design. It will reverse the whole process, and it will learn how to learn the distribution of the noise, and step by step remove the noise and construct a brand new structures and maybe never exist in the real world. So in this case, we can use this whole process to just generate as many as peptide we want. But here bring another question to us. So we can generate hundreds of thousands of peptides, but how can we evaluate the property, like the quality of the peptide? Let's bring another challenges to us. So here we embrace this pipeline released by recent advancements from Baker teams and other teams. They released the 3 science paper last August, and the whole pipeline is use 3 major AI models. First, they use the Rosetta field diffusion to generate a backbone. So in this case, it will have the alpha carbon for the amino acid, but in this case, they do not have the side chain. So they additionally use the protein message passing neural network to implant the amino acid sequence into the backbones. And then they will generate thousands of hundreds of thousands of these sequences for the for the peptide. But how do you know which one is good? How do you know which one is bad? So they introduce AlphaFold 3 to evaluate the whole structures. So in this case, we can evaluate quantify the qualities of our generation. So the basic idea is use this pipeline and apply on this benchmark high quality structure of TCR peptide MHC, and we actually extract the 10 high quality crystals. But for our current study, we only focus on the MHC class 1. In this case, you will find that all the peptides have the same length of dimer. Since this is a comparison analysis, so we would like to have all the shape in the same phase, so we can evaluate them. So to have this diverse benchmark comparison, so we have different allele type. So we will say that some is from the allele type A and B and C. We end up select one allele E from this PDB complex. So bring the initial step to our analysis is how can we add the constraints to our complex. So here I bring a example. So this is the allele type A02 from the peptide MHC complex 1. And for this hotspot analysis, I will say the anchor point analysis, you will say that the second amino acid is located predominantly in the B pocket, and the end of the amino acid is predominantly located in the F pocket. So in this illustration, you will say, so if we can constrain the second amino acid and end amino acid of the peptide, then we will fix this backbone of this peptide into this TCR receptor and MHC complex. And then we can navigate the AI models to implement the whole sequence and backbones for this peptide. And for this illustration on the right, this just remind you this molecular level of the TCR MHC complex. Our goal is to generate the peptide highlighted in this black box. So additional things that's because this beta-2 microglobulin is invariant part, and they have the same sequence. It's actually the same shared across different allele type. So, and additionally, from this figure on the right, you will see that it has no contact with the peptides. So for our entire diffusion process, this beta-2 microglobulin will be dropped. So we will only focus on the interface of the MHC alpha-1, alpha-2, alpha-3 chains, and focus on the CDR3 loop. CDR3 loop is highly variable regions on the MHC, on the T-cell receptors. You will see this. This is the CDR3 loops from the TCR alpha, and this is the CDR3 loop from the TCR beta. So with these contact regions, we can actually define different contact points. So our experiment setting here is a combination of the anchor points, contact regions, anchor points in the MHC, and the contact regions between the peptide and the TCR receptors. And you will see that the purple color is from the pockets B pockets from the MHC, and the light blue color is from the F pocket from the MHC. With this different settings, fix the backbones within the B pocket and F pocket. And we have defined additional information of the contact regions for the T-cell receptors with the peptides. So this whole eight different experiment settings can provide us with comprehensive analysis on how can we navigate the models to generate the high quality of the peptides in the T-cell receptors and MHC interface. So this is our proposed innovative part for this 4-stage filter pipeline. So the workflow is starting with a single template. So we remove the peptides in the crystal structures from the existing PDB database. And the first step is generate 320 thousands of candidates or sequences. And then we will use additional 3 additional filtering steps to filter out the generated peptides with low quality or low scores quantified by our models. And you will see that for our innovation part of this filtering stage at Sigma 2 is that we group the different tools together and provide this consensus ranking to select the high quality of the peptide. And for the remaining step, it's more like apply the generative peptide with the model, evaluation model like the net MHC pan to evaluation the bindings with between the peptides and MHC, and using the AlphaFold evaluation metrics like IPAE and PLDDT to return the quality scores for the entire complex. So if this score is higher, then that means the complex is in high qualities. So I will navigate you through this second stage of physical filters divided by our whole team. So we basically desired two tracks. So for the first track, we feed the generated peptide into the fold X and extract the properties returned by fold X. And we build this logistic regression classifiers based on our existing database, binder or non-binder database, and predict if this generated peptide will be recognized by the TCR receptors. And the higher probability indicating there is a high chance they are binding. So low chance, lower value indicating this this not binding. So we will mark as non-bindings. And for the track B, we feed the complex with this OpenMM. Basically, we went through the molecular dynamic estimations, and then after the short amount of the molecular dynamics, we will extract the Rosetta fold delta G values, and when we will rank this binding probability and the delta G values. So for your information, if the delta G value is smaller, then that means there's strong potential that this complex is highly reliable. So based on these two metrics, we can get two rankings, and we use the consensus ranking basically to get the geometric mean of these two rankings, and we end up with top 100 high quality results for our whole pipelines. And then we will use the AlphaFold 3 evaluation to evaluate this top ranked peptides. So talking to our results, so you would wondering how did this generative model actually perform? So you will see from the table 2, and we list the metrics we extracted, and we average them, get the standard deviation across the two models from the Rosetta fold diffusion 1 and the Rosetta fold diffusion 3. And you will see that the RF1s constantly outperform the Rosetta fold diffusion 3, which is out of our expectations, because Rosetta fold diffusion 3 released last December, just like a couple of months ago, and their team states that this is the state of the models for the protein designs. But I guess because this model is mainly trained for the mimic binder, protein mimic binder designs, so they are not desired for this peptide designs. So the original Rosetta fold diffusion 1 have the higher performance than the Rosetta diffusion 3. This is our the initial guess. So further investigation is undergoing. So we would like to say if this really AlphaFold diffusion 1 has a real best performance for this domain. So if in this case, in our future pipeline, we will adopt the Rosetta fold diffusion 1. But for only one matrix in this table, you will notice that the Rosetta diffusion 3 actually have the higher binder probabilities for the generative models. So that means this Rosetta fold diffusion 3 is more it's more likely to explore this novel conformational landscape, and it's highly enriched for the futures to promote or enhance bindings. So in this case, the Rosetta fold 1, Rosetta fold diffusion 1 focus on the structural evaluations, but Rosetta Field diffusion 3 is prefer to give you the binding probabilities for your generated complex. And on the right table, you will see still overall for this 10 selected PDBs, the Rosetta Field diffusion 1 outperform almost all the evaluation metrics comparing to the Rosetta Field diffusion 3. So we will continue working on this to provide additional evaluations on the remaining high quality PDBs and to give you a comprehensive analysis of this benchmark. So this work is still undergoing, and I would like to bring your attention to the next part. So this is a combination of the different settings, as I just mentioned, that we actually have 8 different experimental settings for the hotspot analysis. We select some contact regions on the MHC, also have the selections on the TCR receptor CDR3 loops. Based on this different constraint, we would like to say if we provide more constraints on the structures, will it perform better or worse? So based on this table and the chart on the right, you will see that we actually find the optimal regions, optimal zones. Like if we define the hotspots, like the contact regions between the number of 12 and 16, the overall root mean square deviations for the generated peptide is smaller, and the predicting binding probability is much higher than the other regions. So for the real design, that's if we conduct the analysis with wet lab validation, we probably will stick to these findings to define the hotspot within the region of 12 to 16 hotspots in the interface of MHC and the TCR receptors. Talking to the final results, and just remind you of this whole pipelines, as we use the Rosetta Field diffusion to generate the backbone, and then use the protein massive passing neural network to impact amino acid sequence into the backbone. And then we use AlphaFold 3 to evaluate the whole complex. So in this case, if we have one template as input, we will generate like 320,000 sequence, and then go through different evaluation process. We end up with a 20 complex. That is, we believe is reliable and in high quality, and that is worth for the wet lab validations. So if we go through this wet lab validation, we project that we will have 20 to 30% of the accuracy. This is the true by this peptide will be recognized by the T-cell receptors, and also they will fit into the MHC. So for the entire benchmark analysis, focusing on the dry lab, but this is only first part of our study. The next ongoing efforts will be focused on the wet lab validations, and which will not release by today's presentation. So additionally, you will have some interest is how this model generates the peptides. So on the left side is original visualizations of this PDB AGOM with the dimers. And you will say that with the AlphaFold diffusion 1 and the AlphaFold diffusion 3, the generated peptides is almost like the identical with the original shape. So you will say that, so this indicating both of these two models can generate a high quality peptide and very close to the original ones. And this brings the end of our limitation of future directions. So our benchmark is basically rely on the idealized settings. So we extract the high quality structures from the PDB database. And this is not the real settings. So in the real settings, we probably only have the sequence information from the T-cell receptors. And so we probably need to generate the shape for these T-cell receptors, and we need to perform this flexible dockings to say if the T-cell receptor can have the which docking angles is the best for T-cell receptor and MHC, so they can recognize the peptide. So this is ongoing effort, and we're actually currently working on this part. And the second part is that this study is most focused on the MHC1. which will actually provide the additional results for the MHC class 2, and which haven't released in our archive papers, but in the updated versions, we have input this information in for the MHC class 2 result. And for the things you might notice that for the entire pipeline, because we only have the constraint on the B pocket and F pocket, that means from the second amino acid to the end amino acid can be best designed by the AI models. But how about the first amino acid? So actually, you will see that entire results only have amers. So that means the list of the A amino acid. We still lack the first amino acid because this constraints cannot provide the high quality on the first amino acid. So this is the first stage. And second stage, we actually need to go to the motif scaffoldings and partial diffusions, as we can have append the first amino acid on top of the amer, so we can complete this peptide generations. So this is our future directions. And additionally, is we would like to iteratively refine this pipeline so that we would like to achieve the high binding chance for our desired peptides in the TCR receptor and MHC complex domain. So for the application, it's just one example. This is actually the MHC class 2, and you will see the binding pattern is very different compared to the MHC class 1. So we will have 4 hotspots, our 4 anchor points in the MHC group. So we are actually working on this real case. So for the multiple sclerosis in the autoimmune disease, these T-cell receptors for the OB1A12 is actually our target. So we would like to design the peptide that's self peptide can be recognized by these T cells and be presented by the MHC group. So in the future, we would like to release this dysfunction self peptides that's cause autoimmune disease. So this is our final goal, and this is actually the application of our pipeline. And I believe that brings the end of this presentation. So if you have some questions, I'm happy to take any question you have during this presentation. Thank you.
Thank you, Dr. C. Thank you very much. There is a question, of course, but I want to first ask, maybe before we go to the available question, I would like to understand the entire workflow where we start from when we want to generate the TCR MHC. Like I see we are using the RF diffusion, both
Yes.
1 and 2. But what is the starting point where the input into the diffusion model in order to come up with the peptide at the other end? So I want to understand about the input data at that stage initially.
Okay.
Yes.
So let me focus on this complex. So our entire input data is coming from the PDB database, and this is crystal structures. And we have the structures for T-cell receptors, structure for the MHC group. And our goal is to generate a peptide. So we remove this raw peptides inside the complex and put this whole structures as input data, and then define the hotspot like the B pocket and F pocket on the MHC and the T-cell receptor contact regions of the peptides, and ask the model and to generate the peptide. So this is the start point for our model.
Okay, that's great. So there is I think there are 2 questions here. One, how do generative models account for the structural constraints of TCR-P MHC binding?
So the constraint that's more like you will see that can you see my mouse over here?
Yes, yes.
So if this is a peptide there, actually the P2 is actually located over here. I mean the B pocket is located over here. So we extract the amino acid sequence on this MHC on the left side and the right side. So they have the close contact with peptide. and use this as a start point for and also as the constraint in the Rosetta diffusion. So it will actually generate the peptide with the constraint domain. So from the P2 to Pn. So you will generate the peptide backbones between the B pocket and F pocket. So basically, you will generate a whole chain of these things, but they will have different, more like this different conformation shape of these backbones.
Okay. Then another question is, how do current generative methods compare in terms of binding affinity, specificity, and structural stability?
So that's actually the part we're focusing on this stage. So for the binding affinities, we basically we trained logistic regression models on our binding or non-binding database and feed these generation models into this classifier. And this will return the binding or non-binding evaluation metrics to us.
Okay.
And additionally, sorry, I got someone knocks the door. Yeah. Additionally, for the reliability analysis, you mentioned that. So we actually went through the molecular dynamic analysis for a short period of amount of time. And after this molecular dynamics, we will extract the matrix for the delta G, and this delta G can reveal the stabilities of the whole complex. So you will both have the bindings and the stabilities based on these two combined evaluation metrics.
Okay, thank you, Doctor. Mine also is about one of the limitations you highlighted is you're intending to work on, I think, motif scaffolding.
Yes.
And my assumption is, or my understanding, correct me if I'm wrong, is the motif scaffolding would help to sort of like predict stability within the peptide.
Yeah.
And so what can you say about, in the absence of motif scaffolding, what can you say about the stability of the designed peptide with this one?
Yeah, this bring the challenge to us as actually our first stage will only generate the 8 amino acid, but the target is 9 amino acids. So that's why we need embrace the scaffold, motif scaffoldings and partial diffusions. Both will cover this the first location region for the B pocket and will generate the P1, the first amino acid of the peptide, and will append it to our previous generations. So that's the reason we need this motif scaffoldings. But in terms of the binding probabilities, so we will have the second stage evaluation. So the first stage is just evaluation of the AMER generation. The second stage will evaluate the whole process, the complete version of the peptide. So basically, that's why I mentioned that because it's unpublished pipeline, so I cannot provide you the whole pipeline into this presentation. But after recording, I can show you the slides. That's actually our whole process is two stage. The first stage is what I presented today. The second stage, I will review it later on.
Okay. Doctor, you talked about hotspots and
Yes.
and I'm wondering, this is sort of a naive question, but it's to deepen my understanding.
Yeah.
What exactly are these hotspots for, and why should we care about them? Like, I know it might be probably connected to binding to the resulting sort of like maybe protein or peptide or antigen, anything. But at the core or a deep understanding, maybe for everyone else, why these hotspots at the deeper level?
Yeah, so the hotspot is the notations solely from the Rosetta for diffusion. So in the real world, for the TCR receptor MHC complex, we normally call this called as anchor point. So the anchor point is actually what I'm described multiple times in this presentation is B pocket and F pocket. It's actually the anchor the whole pipeline backbones into these two locations. So they fix them so that you cannot go anywhere. Just fix over there and to do the backbone generations. But return to the Rosetta diffusions because you cannot define a start point on the unknown locations. So you can only start the diffusion process with known locations. So this known location is actually noted as hotspot in the Rosetta flow diffusions. So we use this start point. How can we define the start points? So basically, we just locate the B pocket and F pocket on the image groove and use this B pockets, have the close contacts with the amino acid sequence on the MHC and use that as hotspot. So that you can navigate the model to generate backbone. So basically this is two notations, but the same things. So hotspot is the locations in the MHC, and the anchor point is actually the 30 space regions that's defined in the fixed point for the peptide.
Wow, thank you very much. One thing as I'm looking at this work is one of the things we struggle with as we are trying to find our interest is computational resources, but also like computational resources, I would say. And I'm wondering for low resource labs or groups that don't have computing power, how can you scale, or not scale, but project the kind of computing resources they might need to run? I know like RF diffusion, I think, needs a lot of computing power, but what could be the computational needs to sort of pull off this kind of work?
So to make this study available, we need to have a multiple GPU card. So for instance, our lab have 8 H200 GPU card. So that's why we can make this happen. But bring you for your attention and your low resource queries. Actually, I learned that NVIDIA have released a kind of the research program. As basically, you need to write a proposal to there, and they will release 2 GPU card to you if you figure your research plan is reasonable and you guys need this GPU, and more likely you don't need to pay, and you will get 2 GPU card. And I think that 2 GPU card can be fully used to replicate our analysis, but might take amounts, I would say 2 months to fully replicate this benchmark analysis. And for the real settings, might spend you around 2 weeks to generate the high quality backbone or high quality peptides in this MHC TCR MHC.
Okay. I think the last one for me is about the protein MP, the message passing.
Yes.
I think neural network.
Yes.
And I think somewhere you highlighted how it was used to design, yes, the pipeline to obtain the sequence. So I'm trying to understand its role here in this work.
Yes.
And yeah, if you could highlight that.
Yeah, so more like this is 3-stage process. The first process is the Rosetta flow diffusion can only return the alpha carbon for each amino acid. So they will not provide the side chain information for the Rosetta flow diffusion 1. But for the recent released Rosetta flow diffusion 3, they actually integrate the protein message passing into the diffusion process. So they can generate the a side chain for you. So that means you will generate the whole sequence for this backbone. So more like they merge 2 steps into 1 step. But ideally, they will implement the protein message passing neural network for this whole pipeline. So more like step 1, generate backbones, lose sidechain information. You need to use protein MPN or ligand MPN, or maybe the Hermers, like the recent models, to impact the sequence for the backbone, and just provide the sidechain information. So this is more like different steps.
There is a question here, Victor, which is someone is saying, I've been reading on uncertainty lately and the different modes of uncertainty.
Yes.
So he's asking, how do you quantify and account for uncertainty in the generated peptides from the models and throughout the pipeline?
Um, so our full innovation is bring this pipeline to be public because we integrate three filters, and these different filters will reduce the uncertainty of this uh, generated peptides. So first is use the existing tool and providing the binding affinities between the peptide and the MHC, and as first stage. And the second stage is using the binding probability and the reliability analysis and group them together to get additional rankings, and then use the AlphaFold3 to evaluate the whole complex. So based on this three-stage filter, so I would say the uncertainty will be reduced to the minimal levels. So we will get very high quality generated peptides.
Right. Thank you very much, Doctor. And I want to thank you very much once again. We are approaching the top of the hour, and I don't see more questions from the audience. And if there are no further questions, I want to thank everyone who has attended this session, but
Thank you.
also in a special way, thank Dr. C for saving up time and really share this novel work with us. Doctor, just for clarity, I think I'll take out the slides that have results.
It's all right. So I mean, for this whole entire presentation is it's okay to be public. I haven't showing the final pipeline. So if
Oh,
you're
right.
stop recording now, I can show our full pipelines for some unpublished result.
All right, we still have 80 minutes, so I'll stop the recording.